we can talk about cells. So looking here on our first page, I like to put an image or something on the first page. We just have the textbook version of what a cell looks like. Remember thinking about our levels of organization from our first lecture. We just finished talking about the chemical level, which is where we've got atoms and molecules and all that wonderful stuff. The second level is the cellular level, where we're looking at cells like what's shown here. And inside of a cell, you can see is a very intricate arrangement of structures called organelles, like the nucleus and the mitochondria that I was just talking about, the Golgi complex, and so on and so forth. What we're going to do is learn about each and every one of those, which should be a review, whether you're taking anatomy with me and you learned about those last week, or whether you have to think back to like high school biology or another college class that you've taken. First thing I want to do is just define and talk about a cell as a whole. We can describe it as a basic unit of the human body. In fact, we all began our life as one cell formed when a sperm cell meets an egg cell from mom and dad that unites and forms a zygote. That zygote is the one cell that then divides, and eventually here we are made up of trillions of cells. Textbook definition of a cell is a living structural and functional unit enclosed by a membrane. When discussing cells, it's important to have a decent idea of what the cell theory is. Modern cell theory is one of the most important unifying themes and theories in all of biology, and therefore this class, since this is an offshoot of biology. There's 5 basic principles of the modern cell theory. Number 1, all organisms are composed of cells and cell products, meaning that if it's living, it's made up of cells, and the cells make the products. And that's what makes up an organism. Number 2, a cell is the simplest structural and functional unit of life. So in other words, cells are the simplest things that can do those basic life processes like metabolism, growth, reproduction, differentiation, etc. Number 3 really is the one that relates to what we talk about here in this lecture. At least it relates to it the most. An organism's structure and functions are due to the activities of its cells. In other words, what we are and what we're capable of doing as a human being is directly related to what our cells do. For me, talking right now, it's because of various muscle cells in my larynx, and contracting your skeletal muscles in your arms involves muscles and the cells of those muscles in your forearm. Processing what you see, what you hear, comes by way of neurons up in your brain and extending down into your arms and legs and everywhere else in the body. So anything that's happening, you can thank a cell for doing it. Okay, number 4, cells come only from pre-existing cells, meaning they don't just appear out of nowhere. And cells of all species have fundamental similarities. We even are comparable as far as our cells go to simple things like bacteria. One key feature that you find from organism to organism, as far as cells go, is that they're small, microscopic in size. And if you've never wondered why, I'm going to tell you anyway. Why are cells so small? Well, there's 3 kind of factors that limit the size of a cell, and it involves the following: the requirement for an adequate surface area to volume ratio. We're going to talk about what that is because that's the biggest limiting factor. Okay. Well, this one right here is going to relate to number 2 and 3. Okay. Because having an adequate surface area to volume ratio is going to affect the rate at which molecules diffuse. Diffuse means for stuff to come in and out of the cell. And then number 3, the need to maintain adequate local concentrations of specific substances involved with cellular processes. So like in our last lecture, we were just talking about oxygen being used to make ATP aerobically. Okay. Well, for oxygen to go into mitochondria, those molecules have to first come into cells. Okay, so cells need to bring in certain substances and they need to release certain substances. That means they have to have adequate rates of which molecules are diffusing And that all relates to number one. So number one affects number two, which affects number three, or you could kind of say it the opposite direction. So of those three things, surface area to volume ratio is the biggest limiting factor. The problem of maintaining adequate surface area arises because the volume of the cell will increase with the cube or third power of the cell's length or diameter, whereas the surface area increases only with the square or the second power. Okay, so let me explain what that means because it's kind of hard to follow, I'm sure. When we talk about surface area, we're talking about the amount of like plasma membrane, the outer boundary. So like think of it like the amount of wall space in the room that you're sitting in. Okay. The volume is the amount of space on the inside. So like in the case of the room that you're sitting in, it's how big the interior of that room is. So comparing the amount of wall to the size of the interior of the room. Okay. Looking at these two cells, I can kind of show you exactly why cells can't be big, like this one right here. Okay, but these are nice and cube-shaped cells, so they have a width of 10 microns and a height of 10 microns here. But after growth, they go to 20 by 20. So you guys can hopefully all agree with me that this cell is half as large as this cell, right? Okay. Well, it says that they're 10 by 10, but really you could describe them as being 10 by 10 by 10 and 20 by 20 by 20 because they're three-dimensional structures and they've done a good job at illustrating that here. So they're 10 microns tall, wide, and in depth. Okay. Well, as a small cell, you calculate the surface area as 10 times 10 times 6, length times width, and then that there are 6 sides. And that gives you surface area of 600 square microns. Okay. When the cell doubles in size, its surface area does not double up. It actually quadruples. It goes from 600 square microns to now 2,400 square microns. So instead of going up to 1,200, which is double, it's gone up 4 times. Okay. More importantly is what happens to volume. As the small cell, the volume is 10 times 10 times 10, 1,000. Now, the volume doesn't go up 4 times. Instead, it goes up 8 to 8,000. That's 8 times. So what's happening is the inside of the large cells getting super big to the point where the surface area is not able to keep up. Okay. The surface area, the membrane surface area, is where stuff comes in and out of the cell. So it'd be like if the cell got so big that the center of it was not able to get enough oxygen, was not able to get rid of enough of its waste. And so the inner part of the cell, because it's not getting enough nutrients and removing its waste, it's going to start dying off. So in order to not have this happen, cells just stay small. Does that make sense? It's kind of hard to visualize, but that's the reason why. Okay, now with that said, we can actually start talking about the parts of a cell. First thing I want to talk about is what we call the cytoplasm and the cytosol. This is the stuff that's found on the inside of the cell. A cytoplasm is a thick gel-like substance made up of various organelles, things like the mitochondria and the ribosomes in the endoplasmic reticulum. And then it also includes molecules that are suspended in a semi-fluid substance called cytosol, or what we've seen already this semester, intracellular fluid. Okay, so cytosol and ICF, as we've seen that abbreviation before, are the same thing. Okay, cytosol and cytoplasm are technically not the same, although you might hear me kind of refer to them as the same thing. Cytoplasm is the jelly-like fluid plus the organelles. Cytosol is just strictly the fluid part. Okay. Although it varies in composition and consistency from one part of a cell to another, cytosol is predominantly water, about 75 to 90%. Plus, it's got dissolved and suspended components. And those things would be like ions, sodium, potassium, chloride, glucose, amino acids, fatty acids, proteins, lipids, ATP, and waste products. A lot of the stuff that we were just talking about in our previous lecture. Okay. One important thing about our cytosol, it's the site of many chemical reactions that are required for a cell's existence. Okay. In fact, we just talked about anaerobic ATP production, where we can make a little bit of ATP without the use of oxygen. That takes place in the cytosol. Okay. For many of our cells' chemical reactions, they're going to take place inside of these, what we call organelles. Okay. Organelles can be described as tiny specialized structures within a cell that have characteristic shapes. You can think of these like tiny little machines that a cell has. Okay. They perform specific functions like cellular growth, maintenance, and reproduction. Despite many of the chemical reactions going on in a cell at any given time, there's little interference among them because a lot of those reactions, as I just said, are confined to our different organelles. Okay. Each type of organelle has its own set of enzymes. Once again, proteins that help catalyze chemical reactions that carry out the specific reactions and serve as functional compartments for specific biochemical processes. Numbers and types of organelles will vary from cell to cell depending on what that cell's job is as far as function goes. Although they have different functions, organelles often cooperate, once again demonstrating the theme of this class, to maintain homeostasis or balance. Okay, we're going to start with our first organelles. And for those of you who are in anatomy, this is, a lot of what you'll see here, word for word, exactly what you learned in that last lecture just the other day. But that's not a bad thing. All right, the cytoskeleton is an intricate three-dimensional array of proteins that permeates throughout the interior of the cell, helping to give it its distinctive shape and high level of internal organization. You see that prefix cyto, that's just a fancy term for cell. So in other words, cytoskeleton is the cell's skeleton. Okay, like our total body skeleton with all those various bones put together, that gives us our shape and helps support and protect. The cytoskeleton does the same for things at the cell level. Okay, three components make up the cytoskeleton. The first, which are the largest, are microtubules, which are hollow cylinders with an outer diameter of about 25 nm. That's short for nanometers. Very tiny are nanometers, but they're hollow cylinders, so they're like a tube or a pipe. Thinking like, a PVC pipe that runs through your house to drain water after you flush the toilet and run it down the sink. Okay, hollow on the inside and structured on the outside. Okay, proteins that make up microtubules are called tubulin proteins. So that's kind of handy. Tubulin makes microtubules that are tube structures. Okay, and they begin in organelles called centrosomes, which we'll talk about centrosomes and what those do here in a second. So what do your microtubules do? And this is what I want you to know, these four bullets. They give shape to the cell or help with forming the shape of the cell, aid in the movement of chromosomes. They aid in the movement of, they aid in the movement of organelles. They also aid in the movement of chromosomes when a cell is going through cell division. And they form specialized cell projections, which we'll talk about here in just a couple of slides. And I would say they're most well-known and easiest thing to see them doing is helping with moving chromosomes when a cell divides. We'll talk about this towards the very end of the lecture, but if you've ever learned about mitosis and meiosis, there's a phase when these things that look like spider legs come to the center of the cell and grab and pull the duplicated chromosomes to the opposite ends. Those spider leg looking like structures are microtubules, and that's how they play a role in cell division. Intermediate fibers or intermediate filaments are thicker than microfilaments, but they're thinner than microtubules, composed of several different types of proteins depending on what that cell's function is. Okay. Overall, what they do is help resist mechanical stress to the cell, stabilize the position of organelles, and help cells attach to one another. And then the smallest are microfilaments made up of a protein called actin. What they do is help generate movement, like during muscle contraction, cell division, cell locomotion, which is where cell can move, Provide for much of the mechanical support responsible for basic strength and shape of cells. So they, have a big role with strength and shape, and provide mechanical support for cellular extensions. Speaking of cellular extensions, let's talk about those here on this slide. Now, a cellular extension and a cell projection, those are the same thing. These are just parts of the plasma membrane that are extended outward to give special function to a cell. If I go back to our very first page where we've got our picture of the cell, you can see, it's a fairly round ball-like or spherical cell, but they do show a few of these kind of random extensions that are pushed outward from the plasma membrane. And the three ones that I want to talk about now, microvilli, flagella, and cilia, play a specific role in the body. Microvilli will be found on epithelial cells lining our intestines and other areas where absorption is important. Okay, what absorption is? The movement of substances into a cell. Okay, now the word microvillus is the singular form of microvilli, and since only one is labeled, that's why it's villus instead of villi. Okay, but they are just extensions of the cell, like little fingers that enable the cell to have increased what we call surface area. Okay, in other words, there's more plasma membrane, more wall of the cell, and the plasma membrane is where you're going to find the doorways for stuff to come in and out of the cell. So by having more plasma membrane, you've got more doorways, which is why they're great at performing absorption. Okay. At the core of them, you'll find bundles of microfilaments, which we just talked about on the previous page. Okay. Cilia are hair-like processes about 7 to 10 microns in length, composed of microtubules. Their job is to move in a rhythmic coordinated way to push substances along the surface of the cell. Looking at the figure here on the next page, and these are real pictures of real microvilli, cilia, and flagella. They look like tiny little hairs on the surface of the cell, and what they do is they just push stuff along the surface of the cell, like here in the trachea, which is our respiratory system airway. It's actually our windpipe, most commonly called. When we breathe in dust and other particles, which is inevitable, we have mucus that is found on the inside of our trachea that will collect that dust so it doesn't get into our lungs. But we don't want that dust and the mucus that's been contaminated by that dust just sitting in there. So the cells that have cilia that line our trachea are going to push that mucus upward back into our throat, or we can either cough it up, spit it out, or what we do more of actually is dump it into our esophagus to be carried to our stomach for digestion. So we're actually kind of digesting dirty mucus all the time, but it's really beneficial and it's a really natural thing. Just sounds kind of gross. Now, on the right is not an illustration, but an actual picture of flagella. Now, flagella is plural, flagellum is singular. These are long, fine, thread-like organelles protruding from the cell surface. They usually move an entire cell by generating forward motion along its axis by rapidly wiggling in a wave-like pattern. If you've never seen, video of sperm swimming, they look like little snakes swimming in water. Okay. Difference between flagella and cilia. They both move, except the difference, flagella actually move the entire cell. So the cell can swim up and fertilize the egg, which happens up in the fallopian tubes. Cilia just moves stuff along the surface of the cell. So the cilia just acts like a conveyor belt, moving stuff along. The cell just stays put, though. Flagella, they allow the cell to entirely move. Okay, centrosomes. If I go back to our slide here on the cytoskeleton, said that microtubules begin in organelles in a centrosome. So what are centrosomes? They can be described as organelles located near the nucleus. Structurally, they consist of two centrioles. You can see 1 and 2 there, and pericentrial or material or matrix. Stuff I'm not really going to get into. It's all kind of real small, three-dimensional, pretty complicated stuff. What I want you to know are the 3 bullets as far as what centrosomes do. So these last 3 bullets would be what you want to star or highlight. Their job is to function to anchor and assemble microtubules, aid in the movement of the chromosomes during cell division, which, like I said, we'll see later on in the lecture. And then those flagella cilia that we just talked about, they help with moving those. So not a whole lot, but centrosomes are important for those kind of 3 things. All right, that brings us to the control center of our cell. The largest organelles of our cells are its nucleus. And generally, a cell only has 1 nucleus, serving as the cell's information or control center. Surrounding the outside of the nucleus is its own wall-like structure called the nuclear envelope, or nuclear membrane. You can see it here on the next page. It's a double membrane that just keeps stuff in and out. But it's not gonna just prevent anything from leaving or coming into the nucleus. And that's because there are what we call nuclear pores present throughout the nuclear envelope. Those allow for stuff to come through either in or out, but with specificity. Not just anything can come in and out. It's specific things. Something like RNA actually is able to exit once it's made inside the nucleus. And I'll get to more about that in a second. Let's talk about DNA, not RNA. DNA is where you will, or is the important thing that we just talked about in our last lecture, responsible for containing the instructions for making our proteins. And in the nucleus is where you find our DNA. Now, depending on the state of the cell, you're either going to find DNA in the form of what's called chromatin, like you show here in this picture, which is showing you nucleus zoomed in 10,000 times. Or if the cell's getting ready to divide, DNA winds itself up, and this is the process in which that happens. And we find our DNA in the form of chromosomes. Most of the time, this is what you're going to find the nucleus looking like, with the DNA unwound, uncoiled up in the form of chromatin. It looks just like a grainy mass. It's not just DNA. There's other stuff in there, proteins and RNA as well. But just before the cell divides, it needs to duplicate its DNA and then wind itself up. It uses these various proteins called histones, and then it forms these they look like capital H's or capital X's, and what we call a chromosome. And now chromosomal number is very important. When a sperm meets an egg, the 23 chromosomes from the sperm, 23 chromosomes from the egg unites, and that creates a cell with 46 chromosomes. Most of our cells contain that same number, and I say most because we produce sperm for us guys that have 23, and women produce eggs that only have 23. That way, when we produce offspring, they're genetically half us and half our mate if we make babies. So what is a chromosome? It's just a long winding molecule of DNA, and what you're going to find on a singular chromosome are thousands of genes. Genes are the basic units of heredity. Now, if this was biology, we'd spend a lot of time talking about this, but each little segment, and there's going to be thousands of them, codes for a different physical trait or a different protein. So like keeping in line about talking about skin color, maybe this segment of this chromosome is going to code for what type of melanin you produce. And And in doing so, that's what controls your skin color. Okay, maybe over here on this side, this segment right there, maybe that's going to code for your eye color. Okay, we have 23 pairs of chromosomes, and each chromosome contains thousands of genes. So we have a lot of different, physical features and different things that our chromosomes do for us. Okay, all of our genes contribute to our uniqueness and why we're all different from one another, unless you're an identical twin, which we'll talk about that later on the semester. Now, overall, what does your nucleus do? It controls all cell activities because it contains the DNA that has the instructions. It stores our genetic information, that's the DNA. And then we synthesize DNA from DNA. That's what we have to do before the chromosome actually, or when our cells divide and we split chromosomes. We also synthesize RNA, and that takes place inside of what's called the nucleolus. This is a non-membranous structure within the larger nucleus. So looking here, this entire ball-like structure is a nucleus. The center of it is a nucleolus. It's like a second organelle inside the bigger organelle. Okay, I would highlight this. It functions to synthesize RNA from the DNA template. We then use that RNA to combine it with proteins to form subunits. We will later combine and form organelles called ribosomes. And we're going to talk all about that here in a little bit. Okay. I've already kind of talked about the nuclear pores and all that stuff. I'm not going to read through the rest of this, but just talks about the kind of doorways that allow things like RNA that's made here to leave the nucleus. Okay. Okay, our next organelles, very tiny little organelles, but very complex, are the ribosomes. We can describe them as the protein factories. In other words, they perform protein synthesis. Making them up are more than 50 small little proteins and an RNA called rRNA or ribosomal RNA. Kind of gives it the backbone. If you want to kind of simplify its structure, it's made up of a large subunit and a small subunit. And you can see when it's together, it's a completely functional ribosome. Where do we find them? In many places throughout our cell. If I go back to this previous page, if you look, you see all these little white dots on the outside of the nuclear membrane. Those are all ribosomes. We also find a lot of them attached to the rough endoplasmic reticulum. Okay. Here is a picture showing you just, this is the outside of the nuclear envelope. You can see there's some of those nuclear pores that we just talked about. And then you see a lot of these little dots. Those are all ribosomes. Okay. Now, the proteins that are synthesized by our ribosomes can eventually form new organelles, become part of the plasma membrane, or will be shipped out of the cell altogether. And thinking about all those different functional types of proteins, structural and catalytic and transportation type proteins. We're constantly making proteins. In a lot of cases, we're just shipping them out of the cell altogether. Okay. You also will find ribosomes just kind of free floating throughout the cytoplasm, unattached to anything else. So we call those free ribosomes. There's also ribosomes inside the mitochondria. We literally call those mitochondrial ribosomes, and they make proteins specific for the mitochondria. Okay, our next organelle is what we call the endoplasmic reticulum, or ER for short, a network of tubular membranes and flattened sacs called cisternae that are interconnected. And looking at the picture on the next page, we are zooming in at this little segment right here. Okay, this is our nuclear envelope, so that tells us this is the nucleus. We're not really focused on it so much as what's surrounding it, the rough and the smooth endoplasmic reticulum, or ER for short. Okay, and you can see its structure. Basically, it's just a bunch of flattened sacs with little spaces. Each space is called a lumen, and as you can see, there are lots and lots of ribosomes attached to the RER. Okay. Now, the RER is short for rough endoplasmic reticulum. Gets its appearance and name rough because it's studded with those ribosomes, especially on the side of the membrane that faces the cytosol. And since ribosomes, as we just learned, make our proteins, we can say the main function of the RER is just to synthesize proteins as well. So it just helps those ribosomes make our proteins. The smooth ER has no ribosomes and therefore has no role in protein synthesis. Instead, it does some other things. It makes some of those fats that we talked about the other day, like fatty acids, steroid hormones. You know, we mentioned steroids are a form of fat, and cholesterol, we also learned, is a form of fat. And it's actually a form of steroid. SER is also involved with inactivating and detoxifying potentially harmful substances, things that would be, you know, detrimental to our health, like certain drugs. Now that can be medicinal type or illicit type, alcohol, pesticides, which are chemicals that are, you know, used on crops to, you know, keep the crop from dying, and fungi and, you know, insects from killing it. Carcinogens, those are just chemicals that can cause cancer, which, you know, you've probably heard of, like asbestos before. That's an example of a carcinogen. So the SER is very, very important, and that role as far as inactivating, detoxifying those chemicals really relates to our liver cells. Okay. Now, it also plays an important role in metabolizing our carbohydrates and storing calcium. And in our muscle cells, it actually has a different kind of fancier name, sarcoplasmic reticulum. It's just a different way to say the same organelle. It's responsible for releasing calcium ions, Ca++, which are going to be involved with muscle contractions. And we'll get to that here in a few weeks, about a month is when we'll get to talk about muscles, and we'll talk about the SR at that point in time. Now, what we do with those freshly made proteins from the RER and those freshly made fats from the SER, as I've already kind of said, is a lot of times we ship them out of the cell. But before we can do that, we need to move them so that they can be altered a little bit, which will be done by an organelle called the Golgi complex or Golgi apparatus. We're going to talk about the Golgi here in just a second, but really quickly, I want to cover something very, very basically, and I'm saying basically because I'm going to cover it just at the elemental level, and we call this gene expression. I could spend an entire week or way more than that really talking about this. But I'm going to simplify it. Okay. Although cells synthesize many chemicals to maintain homeostasis, much of our cellular machinery are devoted to synthesizing proteins in large numbers. And that's because, as we know, if something's happening in the body, there's a good chance a protein's doing it, right? So we're needing to make proteins all the time because they do so many different things for us. Okay. So these proteins are going to determine physical and chemical characteristics of cells and therefore us, the organism. Process known as gene expression involves the synthesis of proteins from an original DNA template, and it involves two processes, what we call transcription and translation. Okay. Transcription is where we transfer genetic information from DNA to an RNA. Okay. Or in other words, we transcribe DNA to form RNA. That way, we can then take that RNA and run it through the process of translation, where we involve those ribosomes that we just talked about, the little protein factories. We take our sequence of amino acids, like letters of the alphabet, we put them together, just like we do with words, and we make a fresh new protein, which is going to give us, that specific function. If it's melanin, it gives our skin its color or our hair its color. Okay. Translation can be described as conversion from nucleic acid language to the protein language. Okay. I'm going to simplify expression by kind of writing out the following phrase. It is essentially something as simple as you start with DNA and you make RNA. Then you take that RNA and you make a protein. The process where we go from DNA to RNA, that's what we call transcription. Where we go from RNA to the protein, that's what we call translation. Now, like I said, I could spend a lot of time talking about this. Transcription is very complicated. DNA is a double helix, RNA is a single strand, so you have to kind of do some crazy things. And then to go from RNA to protein translation, that involves a whole lot of craziness where we're putting amino acids together in the correct order. We have to read codons and anticodons and stuff I don't want to really get into in this class. Okay. If you want to learn about that, you can take biology or an even more sophisticated biology like molecular biology. I took that when I was in college, and we learned about gene expression for like half the semester. I mean, it's very intricate, very crazy stuff. Your textbook talks a lot more about this kind of stuff, but I'm going to keep it very simple and leave it at that. And so as a result of gene expression, as we know, we make the protein. And remember, proteins are what do everything for us, just about anyway. So this is obviously super, super important. Okay. Now, once we've performed gene expression and we've made those new proteins, we can then transport them to the Golgi. Golgi complex, Golgi body, Golgi apparatus all mean the same thing. Another stack of membranes with space on the inside. Okay. I like to tell students to think of your Golgi like the UPS or FedEx of your cell. Its job is to take packages like a UPS or FedEx company would, put them together and transfer them somewhere else. Okay, like delivering a package from one person to another for their birthday or whatever. Okay, so looking at our illustration at the bottom, we show the RER here at the top. You can see the ribosomes. The proteins that were made by the ribosomes accumulate on the inside of the RER. What takes place next is a transport vesicle is formed. The transport vesicle, actually, if I go back to the figure here, we can see one of them starting to form. It's where the RER just kind of forms a bubble and breaks free. And inside, you see those little dots. Those would be freshly made proteins that can be then transferred to the Golgi. Okay, the side of the Golgi that receives those freshly made proteins is called the cis or entry face. Okay, now proteins are carried here, so are freshly made lipids, which come from the SER. Remember, the SER makes fats, RER makes proteins. The Golgi processes both. Okay, so we drop them off here. The middle part of the Golgi is where maybe we're going to add a carbohydrate, or we're going to change the shape of that protein. Okay, maybe we're going to make it into a glycolipid or a glycoprotein. And whatever it is, we alter it. So this is like what happens when a package gets its barcodes and its special numbers and tracking numbers and all that stuff. And then when it's ready to be shipped out, That's what the trans or exit phase allows for. And kind of the same thing happens. We pop these bubbles off, and those bubbles will carry the protein or the fat to its ultimate destination. Sometimes, in many cases, protein needs to be released from the cell altogether because maybe it's a hormone that needs to go into the bloodstream. Sometimes we make a protein or a fat that's going to actually become functional for the plasma membrane. Okay, maybe this is a phospholipid. And as we know, phospholipids are those funky fats that help form our plasma membrane. In some cases, we form an entirely new organelle, like a lysosome that you see here. Okay. So I kind of explained, I'm not going to read through that since I just kind of explained it. Once again, key function of the Golgi is to package different molecules together that can be used elsewhere in the cell or the body. Carbs may also be added to proteins and lipids in the Golgi to form glycoproteins and glycolipids, which I've already said. Okay, let's talk about those lysosomes that I kind of pointed out. Lysosomes are organelles about 0.5 to 1 micron in diameter. So they're very small, surrounded by a single membrane formed from the Golgi complex, like we just saw on the previous page. On the inside of that single membrane are as many as 60 digestive and hydrolytic enzymes that can do a lot of breaking down of certain things. Okay. I like to tell students to think of your lysosomes like the cell's garbage disposal. Okay. It can digest and break things down like your garbage disposal can when you dump food down it. Okay. So what can the lysosome do? It can hydrolyze. It can break down proteins, nucleic acids, DNA, RNA, complex carbohydrates, and fats. So all those organic compounds that we just talked about in our previous lecture, lysosomes can break them down. Okay. We can actually digest and dispose of any worn out organelles. That process is called autophagy. Auto is a prefix that means self. Phage means to eat, so self-eating. Like over time, for example, our mitochondria start to break down, and when they need to be removed from the cell, those organelles become enclosed by a membrane formed by the ER, or the ER. And that forms a vesicle, a bubble called an autophagosome. Then that fuses with a lysosome, and those enzymes inside can then go to work. Okay, they can then break those things down to usable things that can be recycled. So recycled stuff gets released back into the cytosol for reuse. So in this way, we're going to constantly replace organelles, and it's not a wasteful thing. Okay. Our lysosomes can degrade foreign material brought into the cell. Something very important in some of our white blood cells, for example. We have white blood cells in our body called macrophages, which perform what's called phagocytosis. We're going to talk about phagocytosis at the end of the lecture, but that's a process. Think of it like what Pac-Man does. He runs around and he gobbles stuff up. Okay, white blood cells called macrophages engulf like viruses and bacteria, and then the lysosomes destroy those and keep our body nice and safe. Okay, our lysosomes can actually destroy the entire cell, a process called autolysis, something that's purposeful, which seems kind of weird, but we can do it. And some lysosomal enzymes can operate via extracellular digestion. So all that stuff I just was talking about was where digestion takes place inside the cell. That would be intracellular digestion. Remember, intra means inside. Extracellular digestion takes place outside. Okay, and this is what happens like when sperm release the enzymes from their lysosomes to help it penetrate an oocyte, which is just a fancy way to describe an egg. So that's what helps with that process of fertilization and making a baby. Okay, our next organelles are peroxisomes. And I also just threw in proteasomes at the bottom. Good. Space, no need to have an entire new slide for just a couple words. Peroxisomes look a lot like lysosomes in structure, but they're actually smaller and they do different things. Another name for them are microbodies, although you'll never hear me call them that. Inside of peroxisomes are oxidases. These are enzymes that can oxidize, which means they remove hydrogen atoms from various organic substances. We have a lot of these in our liver cells, our hepatocytes, fancy term for liver cells. What they do is they generate and degrade hydrogen peroxide, H2O2, which is a potentially toxic compound. So it creates, but then it just breaks it down. Okay, hydrogen peroxide is the same chemical that you can buy over the counter, used as a cleaning agent, used as a disinfectant if you get a wound. Okay, something you can clean with, but not something will just like naturally inside your cells or like in your fluids. Okay, peroxisomes help clean that up. Peroxisomes help neutralize free radicals. Remember we talked about free radicals a couple of slides ago or a couple of lectures ago. Remember those are like the tornadoes in our cells that are very destructive, where there's that unpaired electron. And free radicals we talked about can cause cancer. Peroxisomes help neutralize those. Peroxisomes also detoxify certain drugs and alcohol. So we just talked about the SER detoxifying drugs and alcohol. Well, it's not the only organelle that does that. So does our peroxisomes. And peroxisomes can also decompose, break down our fatty acids. Finally, our last one here on this page, proteasomes, barrel-shaped structures that destroy unneeded, damaged, or faulty proteins by just cutting them into smaller peptides. So remember, we talked about proteins being long chains of amino acids. If you start to have a protein breaking down or no longer needed, you can just chop it up and it's smaller parts, and that's what a proteasome will do for you. Okay, and I believe this is our last organelle, and that is our mitochondria, which we talked about earlier today as we finished up our last lecture. We learned in that lecture that this is where we undergo aerobic respiration, which once again, aerobic means that it requires oxygen. Okay, so aerobic respiration process involving oxygen results in the manufacture of ATP. Nickname for them, you hear it all the time, is the powerhouse of the cell because of their role in making the energy that powers our cells. The more active a cell, like our muscle cells, the more energy they require, which means the more mitochondria they'll have. There is a really unique and pretty cool structure and makeup to our mitochondria. They've got a double membrane, so there's two membranes surrounding it. The outer membrane, shown here, nice kind of orange, and then kind of in the lighter orange, yellowy color, is the inner membrane. Okay. Now the outer membrane is smooth, but the inner membrane, and you can see because it's been opened up, is folded inward on itself, and that's very important. Okay. Those inward foldings have a special name, what we call cristae or cristae, mitochondrial cristae. And this is where you're going to find most of the intermediates that are required to extract the energy from our food molecules. Okay. So in other words, the cristae, and if we zoom in at it, we can see this is where you're going to find enzymes and other structures that are used to make the ATP. If you've ever learned about electron transport chain, the enzymes that you see here are the proteins involved with making ATP in that process. Okay. By being folded inward on itself like this, it increases its surface area, which means you can have a lot more of these enzymes and therefore a lot more ATP production. Okay. Other reaction sequences can take place in what's called the matrix of the mitochondria, a semi-fluid that fills the inside of the mitochondrion. So it's inside the cristae. This is where you'd be going through what's called the Krebs cycle or citric acid cycle, something many of you may have learned about before. You can see little blue dots in our picture there show you that there are ribosomes as well inside the matrix. Talked about ribosomes a little bit ago and how our mitochondria have them. There's also going to be a unique form of DNA here, what we call mitochondrial DNA, which is really cool. This is the only other organelle aside from our nucleus where you'll find DNA. And interestingly enough, we generally only inherit it from mom, not from dad. All right, and that therefore wraps us up on our cellular machines known as organelles. What we're going to do is transition from them to now talking about what we call the plasma membrane, which is the structure that surrounds the outside of the cell, which we actually started to talk a little bit about on our previous lecture, biochemistry, when we learned about those fats called phospholipids. Thinking back to what we talked about just the other day, we said a phospholipid has that kind of unique feature of being amphipathic, where it's got polar and non-polar parts, right? It's got a head and it's got two tails. The head, we said, is polar. The tails are non-polar, just like most other forms of fat. But that influences their relationship with water. We can describe the heads as being hydrophilic, whereas the tails are like other forms of fat and that they are hydrophobic. If you can't remember what hydrophilic and hydrophobic mean, think of it as hydrophilic, they like water, hydrophobic, they don't like water. So smiley face, water, face, water. Okay. Hopefully that all rings a bell. We just talked about that 2 days ago. Okay. It is thermodynamically favorable for the nonpolar hydrophobic acid chains of the phospholipids to face one another and the polar heads to face outward when placed in an aqueous solution. Aqueous solution is a watery solution, which we, as we know, have all over the place. Places like inside the cell and outside the cell, what we call intra and extracellular fluids. Okay. Results in the formation of a two-layered membrane we call the lipid bilayer. And as you can see, phospholipids are very prominent, but they're not the only components of the plasma membrane. This is a very just small little segment of it. You also find other forms of fats, like cholesterol, for example, is even in there. We even find glycolipids, carbohydrates attached to a lipid. You'll also find proteins, okay? Proteins like this one here and this one here. You'll find glycoproteins where a carbohydrate's attached to a protein. Now the ratio between these components, the amount of proteins and the amount of carbohydrates depend upon the overall function of the cell. All right, let's talk about the specifics and what actually make up the plasma membrane. Now, I was kind of earlier in this lecture referring to the room that we're, sitting in, whether that's here in Wamego today or there in Atchison. Think of the room that you're in as like a cell, okay, a structure that's, fairly square with walls, a floor, and a ceiling. Okay, but I don't want you to think of a cell in the same way, because unlike the walls of the room that you're in, the plasma membrane is very not rigid. It's very dynamic, it's very fluid. In fact, the arrangement of those fats and proteins and carbohydrates can be described using what's called the fluid mosaic model, which basically means that things are kind of floating around and easily rotating throughout the bilayer. Okay, so it's not a rigid structure, and it's fluid, it's dynamic. Okay. All right, let's talk about the membrane lipids or fats. As we just saw on the previous page, phospholipids, most prominent of the lipids make up about 75% of the lipids in the plasma membrane. I didn't want to include the same information twice, so that's why I have it listed there as see previous page. Glycolipids are going to represent only about 5% of the lipids in the plasma membrane. We only find them in the layer facing the extracellular space. So in other words, they only face outward from the cell. They help with adhesion among cells, so they help cells like stick together. They may mediate cell-to-cell recognition and communication. So in other words, helping cells recognize one another and communicate. And they also play a role in cell growth and development. And then our third and final membrane lipid is cholesterol, which is about 20% of the lipids in the plasma membrane, but are found only in animal cells. I don't even know why I included that because we're only talking about us in this class. But they strengthen the plasma membrane. At the same time, though, they do decrease its flexibility. Okay, so those were our membrane lipids. Let's talk now about the membrane proteins. We classify them based on their location within the membrane as either being integral or peripheral. Integral proteins are going to extend or through the bilayer. Okay, if I go back to our picture, and what this means is that they can be found throughout the center of the plasma membrane, and many of them are transmembrane. Trans is the prefix that means across or through. So transmembrane go across or through the membrane, so they penetrate all the way through, where there's part of it sticking into the cell and touching the cytosol, and then part of it touching the extracellular space outside the cell. Okay. Now, many of those integral proteins are glycoproteins, which are like lipids, except now it's the carbohydrate attached to a protein instead of a lipid. Okay. Same situation as glycolipids. Glycoproteins will be attached and found on the extracellular side. And so what we can find is that cells have a unique kind of sugary coat on its outside called the glycocalyx, which is a result of both the glycoproteins and the glycolipids. Why this is important? Because each cell has its own unique sugary coat. This carbohydrate pattern is unique to each cell, enabling cells to recognize one another's molecular signature. So cells can recognize one another. It's like if we all had a different color coat on and we wore that same coat every day. And if you looked at somebody from behind and you saw what color coat they were, you'd be able to recognize them. Okay. Cells can recognize each other because of this, which is very important, for example, to prevent like our immune system cells from attacking our own body cells. Okay. It is also helpful for allowing cells to stick to one another, adhere to one another, and protect them from being digested by enzymes in our extracellular fluid. The other type of proteins are peripheral proteins, which are not in the middle of the membrane, but instead are just like attached to the outer or the inner surface. So going back, here you can see a peripheral protein attached to the inner surface. Here's another one on the outer surface. So that's how we can classify proteins, which is mainly structural. We can also classify proteins based on what they do. And this is kind of how we typically describe proteins of the plasma membrane into these, 6 different categories. The first two are two that we're going to talk a lot about later on in this lecture. We're not going to get to that point probably today, since we're just getting short on time. But you've got channels and carriers. Channels and carriers are both integral, so they extend through the plasma membrane. The difference just is between what happens to the protein when something moves through it. Okay, because that's what they do. They allow substance to either come into the cell or out of the cell. Okay, in the case of channels, most of these are going to allow an ion through, so we call them ion channels, and they do so with great specificity or selectivity. So for example, maybe these are sodium ions. This would have to be a sodium channel for that sodium to come in. If it's a potassium channel, potassium is the only thing that can go through it. Okay. And a carrier or transporter, which is what they're also known as, this figure looks like there's two different proteins, but it's the same protein just after what's happened to it, which is where it changes its shape. It undergoes a actual transformation where, like right here, you can see it's got a pore open to the outside. The membrane can, or excuse me, the substance can bond to it, come into that pore, and then the protein changes its shape and the pore opens up so the substance can be released on the other side. Okay. Carriers are, for example, how our glucose comes in and out of the cell. We call those that move glucose transporters. Okay. So like when you went into the room that you're sitting in, whether that was here or there, you know, thinking of a channel like a doorway. I don't know, I want to say door because I'm talking doorway. The entire structure that surrounds the door is like a channel. And like a doorway, there can be a door that covers a pore and a channel. We call these that have a little door right here, opens and closes in response to something. We call those gated channels. And we'll talk a lot about gated channels next week, actually. So we have that to look forward to in our unit 2. Receptors are integral proteins that serve as cell recognition sites. Sites, they allow things to recognize and bind to a type of cell, like hormones that are released in one part of the body that need to bind to a specific cell. The specific cell that receives that hormone is only, or going to be the only cell that has those receptors. And so it's, for example, as you can see, how antidiuretic hormone will only interact with cells of the kidneys and not, cells of the adrenal glands, where it's also going to be. And enzymes, we've talked all about those here the last couple of days. Integral peripheral proteins catalyze or speed up chemical reactions inside or outside the cell. Linkers are integral peripheral proteins anchoring proteins in the plasma membrane of neighboring cells to one another, or to protein filaments inside and outside the cell. So linkers link and hold things in place. And then I've already kind of talked about the last one, glycoproteins and glycolipids. They give a cell its unique coat, acting them or allowing them to act as identity markers so cells can recognize one another. This is important like during tissue formation or to recognize and respond to potentially dangerous foreign cells like viruses and bacteria. Viruses aren't cells, but foreign substances. Okay, I'm going to stop here. This is going to be a perfect place to pick back up with on Tuesday. For those of you who are in anatomy, this is going to be where we kind of really start to get into brand new information. I know a lot of this was just rehashed from what we talked about the other day in class, but here's where things will really start getting more technical and physiological. So we'll start with this on Tuesday and run from there.